plant cell wall is broken by cellulase enzyme. DNA is isolated from the protoplast. The sample is centrifuged, and DNA precipitates as a pellet at the bottom of the test tube. After centrifugation, proteins are removed by protease enzymes. RNA is removed by RNase enzymes. Pure DNA remains in the test tube. Chilled ethanol is added slowly through the walls of the test tube containing DNA. Pure DNA, in the form of cotton-like strands, is extracted using a glass rod.
DNA is extracted in pure form from a test tube. A restriction enzyme, visualized as a baby monkey, cuts it. The enzyme cuts the DNA into different sizes, small and large. This is the work of a restriction enzyme. Agarose gel is poured into a gel electrophoresis tray. A comb creates small wells at the top of the gel. The gel is placed in the electrophoresis chamber. Cathode and anode electrodes are fixed and the set is ready for DNA separation. DNA fragments are taken into the wells of the agarose gel. Electricity is passed through the terminals. DNA fragments move from the cathode towards the anode. Smaller fragments move faster than larger ones. Ethidium bromide is added to the gel. It is illuminated by UV rays. DNA is visualized as orange-colored bands. The required DNA is cut and removed. This is called elution. During denaturation, double-stranded DNA is heated to 95 degrees Celsius. The hydrogen bonds break, and the two complementary DNA strands separate into single strands. Annealing. After denaturation, two primers are added to the exposed DNA templates, one at the 5' end, one at the 3' end. The primer is a short segment of RNA.
Taq DNA polymerase adds nucleotides to the existing DNA templates. New DNA strands are synthesized in the 5' to 3' direction on both templates, following complementary base pairing. Foreign DNA containing the insulin gene is cut at the palindromic sequence by a restriction endonuclease. The enzyme cuts open a plasmid. DNA ligase joins the foreign DNA with the plasmid, forming recombinant DNA. E. coli cells are incubated in calcium chloride solution. The cells become competent to receive recombinant DNA.
The E. coli cell is placed on ice, undergoes heat shock and at 40 degrees Celsius. Pores open, allowing the plasmid DNA to enter. The cell is returned to ice, trapping the plasmid. The cell is now successfully transformed. Microinjection introduces recombinant DNA directly into the nucleus of a living cell using a fine glass needle. The gene gun delivers recombinant DNA into plant cells using DNA-coated gold.
This is the selection of transformants. Transformants are bacteria that have taken up a recombinant plasmid. On an antibiotic plate, only these transformants survive and form glowing colonies. In cloning, a recombinant plasmid is introduced into a bacterial cell. The cell then divides, replicating the plasmid in each daughter cell. This multiplication continues, forming a colony of identical clones. Thank you for watching. It's been a pleasure sharing the wonders of botany. Keep exploring.
Inside the transformed E. coli, the inserted gene becomes active. It is transcribed into messenger RNA. Ribosomes translate the mRNA to produce the desired recombinant protein. After the recombinant protein is produced, it is carefully collected from the host cells. The protein is then purified and processed to obtain the final product ready for medical or industrial use.
Thank you for watching. It's been a pleasure sharing the wonders of botany. Keep exploring.